Hello, everyone. Welcome to our childhood dementia research webinar on brain organoids. It's lovely to see everybody trickling in from the waiting room. We'll get started shortly. I can see we've got 24 people joined now, and I'll kick off because we've got a lot to get through. For those who don't know me, I'm Chris Elvidge and I'm the Head of Research at the Childhood Dementia Initiative. Thanks so much for joining us. Before we get started, I'd like to begin by acknowledging the traditional owners of the various lands on which we are all meeting today. I'm joining you from Melbourne and acknowledge the Wurundjeri Woi-wurrung people of the Kulin Nation and pay my respects to their elders past, present and emerging. In this webinar, we will hear from 4 speakers, followed by a question and answer session. To ask questions, you can type them in the chat box. We'll start our session today with a short clip from an Australian Story episode aired on the ABC in 2018. It shows Stephen Damiani, whose son had a rare form of leukodystrophy, looking at his son Massimo's brain cells under a microscope after he had passed away. We're fortunate to have Stephen with us today, and we'll hear more from him after the clip.
These are the little mini brains that we have made.
It's his way of telling us in a different way to finish the mission.
Okay, well, let's see what he tells you.
Viewing these micro brains today from Massimo's cells now that he's no longer with us, it's very hard to describe. It felt right, it felt good, it felt like we're making progress, it felt like we're continuing on the mission that, Massimo wants us to finish. I feel like it's Massimo talking to us now.
Let's not give up and let's beat this. I know.
I'll have to do that again.
Surprisingly, amazingly, they're still determined to keep that promise.
Okay, so now I have the pleasure of introducing Stephen Damiani. After his son Massimo's diagnosis with a mysterious neurological condition in 2009, Stephen set about learning all he could about genomics. Despite no biomedical training, he then spearheaded an international effort to perform the world's first successful familial trio genome analysis to pinpoint the exact genetic cause of his son's illness. Stephen then established the Mission Massimo Foundation, which is a clinical and research program which promotes the prevention, diagnosis and treatment of childhood leukodystrophies. Sadly, Massimo passed away in December 2017 before a cure was found, but the mission carries on today and is making great strides forward. If you haven't watched the full Australian Story episode, I highly recommend it. It really shows the power of parent and patient-driven research. Stephen will give us his perspective as a parent about brain organoids research. So over to you, Stephen.
Thank you, Chris. Just bear with me a moment to get it all up and running. I think that's all there. Thank you for inviting me to speak today on behalf of Massimo's mission, to share our mission and work with regards to organoid research, also known as the Prometheus mission, which has been fundamental to our efforts in closing the loop from genetic diagnosis through to precision treatment. Before I jump into closing the loop, I just want to take a moment to reflect on how and why I'm here today. The little boy you see here is Massimo, or as we like to call him, Mo, and he also happened to be my son. And back in 2009, without warning, Massimo presented with some alarming developmental regression. His ankles started to stiffen, his toes were curling, and he was having trouble with his balance. He was struggling to pull to stand and he was getting very frustrated. Before long, we found ourselves at the Children's Hospital here in Melbourne, and Massimo was having an MRI on his brain and spinal cord. A new language entered our vernacular that evening, but perhaps the most frightening word we were to hear was something called a leukodystrophy. It was the start of a diagnostic odyssey that took us 1,661 days to solve. So what is a leukodystrophy? Well, the word comes from Greek roots, leuko meaning white, dys, lack of, and troph, growth. Leukodystrophies are a group of rare neurodegenerative disorders that affect the formation of myelin, or the white matter as it's known, in the brain and spinal cord. The prognosis for infant onset leukodystrophies was particularly poor, with life expectancy of only months or at best several years. At the time, there was no cure. Intervention was purely supportive. Tragically, Massimo passed away unexpectedly in December 2017. And Massimo's passing showed us that we could not move fast enough to develop a treatment for this condition. Enter Massimo's mission. In addition to accelerating the discovery of genes for rare neurodegenerative disorders, its objective was to develop adaptable, repeatable, and scalable modelling and treatment capabilities to change the economics for rare diseases like childhood leukodystrophies, and to translate this knowledge into clinical treatments, most importantly, within meaningful timeframes. Massimo's mission is a capability and technology demonstrator blueprint, using HBSL, condition 0 for which Massimo was diagnosed, as a proof of concept disease with Massimo as patient 0. Its objective was to close the loop from diagnosis through to precision therapies for this family of rare diseases. Massimo's mission comprises 4 distinct operations: genomic diagnosis, patient bioregistry, disease modelling, and preclinical testing. Now today, I'm going to be focusing on disease modelling operations, which comprise 3 missions. Specifically, the University of Queensland Prometheus mission, utilising induced pluripotent stem cells to create brain and spinal cord organoids for cellular modelling, led by Professor Ernst Wolvertang and Dr. Mohammed Shekhar. These organoids provide us with a powerful disease modelling capability to test novel treatments using the Genesis mission and the Joint Strike Vector mission, and then hopefully into clinical translation into the Thunderbird mission. The UQ team has created a repertoire of patient-derived cellular models, including both brain and spinal cord organoids from across the disease severity spectrum. Compared with traditional 2D iPSC cultures and animal models, these organoids offer a higher fidelity representation of brain development and function. Organoids derived from patient iPSC lines allow us to study the diseases at different stages and to elucidate the better understanding of the disease mechanisms. Encouragingly, the University of Queensland team has observed defect severity that also matches the patient phenotype severity in both the brain and spinal cord organoid models. In the brain organoids, we've observed oligodendrocytes, myelin defects. In the spinal cord organoids, we've observed motor patterning defects. And in addition, the team has observed, for what I understand is the first time, abnormal motor neuron activity or spasticity, just like what we would see in Massimo in the spinal cord organoids. Remarkably, bathing these organoids in candidate molecules has ameliorated the spasticity, providing an encouraging therapeutic pathway to manage spasticity for improved patient care. Our Joint Strike Vector mission has also recently delivered its first gene therapy payload on the brain and spinal cord organoids, and we're eagerly awaiting the results of this experiment in the coming weeks, which we've worked for the past 5 years to get to this point. Should it succeed, and I have every confidence it will, this will validate an exciting precision treatment for clinical translation into our Thunderbird mission. Furthermore, the team has identified an unexpected avenue to the investigation of a vital non-canonical function of the DARS gene and other tRNA synthetases, which I'm sure you're going to hear a lot more about in the not too distant future, that affects this family of tRNA synthetase affected conditions in the leukodystrophy family. Now, in closing, I'd just like to really make a point here that none of this would have been possible without a confirmed genetic diagnosis. I know we're here today to focus on brain organoids, but unless we were able to identify the cause of Massimo's condition, which at the time was an unclassified leukodystrophy, which we identified was caused by the DARS gene, we can only imagine what a difference those extra 1,158 days would have made if we could have achieved the diagnosis in 72 hours instead of 3.5 years. I take this opportunity to acknowledge everyone involved for how far we've come and thank them for their commitment to invest in how much further we need to go to finish this mission. So thank you for having me.
Thanks, Stephen. You've just made me realise that we need to get you and the researchers back for a full update soon, because it sounds like there's some great things happening.
Thank you. There's some exciting things in the not too distant future.
Yes, I'll be in touch. Don't worry about that.
Yeah.
Thank you. Okay, so now we're going to hear from Dr. Fiona Bright. Fiona completed her PhD at the University of Adelaide, and since then has held various fellowships at the University of Adelaide, University of Sydney and Macquarie University, working on sudden infant death syndrome, early onset dementia, frontotemporal dementia and motor neuron disease. More recently, Fiona received funding from Dementia Australia to pursue research into childhood dementia, investigating links between neurodevelopmental abnormalities and neurodegeneration in the aged brain. Over to you, Fiona.
Thank you, Chris, and thank you to the organisers for the opportunity to present today. So just as a bit of background, I am quite new to human organoid research. My background has been in postmortem human tissue and also animal models. But I see the benefits of utilising human brain organoids, particularly for childhood dementia research, and we're now transitioning our research into these more humanised platforms. So what are human brain organoids? They are 3D self-organising structures of tissue derived from human pluripotent stem cells, and therefore they are physiologically relevant 3D in vitro human models of disease. They're able to mimic the brain's organisation, development and activity within a dish. And this enables us to recapitulate various aspects of the cellular composition, functionality of the human brain across development, but also give us insights into human neurobiology across aging. How do we generate human brain organoids? Well, there are a number of different protocols established in the literature to date. However, ultimately, we always have to start with a source, and that will be either 2D human stem cells taken from blastocysts, or from patients themselves in the form often of skin cells or fibroblasts that can be reprogrammed into any stem cell that we choose to derive it to, and ultimately then produce 3D human organoid structures. In our lab, our protocol is to grow our 2D human stem cells. We then seed them into a plate called an agrawell, which has these small little wells that allow the cells to aggregate together overnight. Then we take up these immature 3D structures and pop them into a dish full of media, and that media contains various different nutrients and growth factors depending on which direction we would like to send the organoid in. And over a process of 40 days, we change those growth factors, and ultimately then mature organoids develop, and in our lab we develop cerebral organoids or cortical organoids. We ultimately then characterize and validate the models and then go on to perform disease modeling and apply various experimental applications. So when first developed organoids are quite immature, the image on the left showing that they have these quite simplistic structures and they're not as layered until they mature over time with those growth factors, and then can have a variety of different cell types and have those layers that then are representative of the cortical layers of the human brain. There is other areas of research in human brain organoids where we can actually fuse together different brain regions, so quite distinct organoids can be fused together, and this can allow for us to look at migrating neurons between different regions of the brain, and they are termed assembloids. So why do we use human brain organoids for childhood dementia research? Well, ultimately, they really are an excellent platform because they simulate the developmental processes and structural characteristics of early human embryonic development. They also allow us to better identify and maintain gene expression patterns and key developmental patterns that are unique to the human. This is really important for childhood dementia, given that a lot of the genetic causes of childhood dementias are extremely rare and unique to that patient. We're able to model the heterogeneity of the brain and its cell populations, regionalization, and the interdependency of specific brain regions. And you can see on the figure on the right, all of the different brain regions of human organoid that can be developed. Organoids can be grown in large number from a very small pool of stem cells, and that means that we have high utility for drug screening and therapeutic development. So I mentioned previously my background has been in human postmortem tissue and animal models. And so these and 2D cell culture have ultimately been what we have built a lot of our understanding on the processes of human neurodevelopment on. These are fantastic models, but ultimately they do have their downfalls. And human organoid models now pose this new platform of being able to be very representative of key unique features of human neurodevelopment. So 2D cultures, unfortunately, are 2D. They don't represent that 3D structure of the human brain. Postmortem tissue is end stage and unfortunately does not give us insights into maybe factors that are associated during life that contribute to the disease. And ultimately, animal models such as mouse models, which I've utilized heavily in my research, they're not human. And particularly during neurodevelopment, this has huge problems because the mouse brain is very different. The mouse brain is small, it is smooth, it doesn't have the invaginations that the human brain does, and it also doesn't have the number of specialized regions. But ultimately, the biggest difference is that the mouse and human brains develop at different rates. Mouse brains are fully developed at 3 to 6 months, whereas human brains don't fully develop until the mid to late 20s. And therefore, human brain organoids are really a much better platform to demonstrate this development across time. So the applications of human brain organoid models, well, the opportunities really are endless. This field of research is booming, and now with greater improvements in bioengineering, there is essentially unlimited platforms and tools to be used to explore human brain organoids in diseases such as childhood dementias. So this is a typical workflow that we put together in our group. So ultimately, you can choose whichever disease of choice that you want to look at. Ideally, we would source patient iPSC lines, just like Stephen mentioned before, because they contain the ultimate foundation of genetic information that's unique to that patient, and we can therefore explore that gene mutation in detail. If we are unable to source patient lines, however, we can genetically modify wild-type healthy stem cells to express our gene mutation of interest with CRISPR-Cas9 technology. We then generate our 3D human brain organoid models. In our group, we've focused on cortical brain regions, but a colleague of mine is now fusing together organoids to look at different regions to explore disease Dravet syndrome. We always characterize our models using a variety of different platforms to ensure organoids are expressing the markers across neurodevelopment. We then high throughput produce them, and ultimately we would like to apply them to drug screening and therapy. So just like anything, there are limitations to using human brain organoids. Unfortunately, because they are 3D structures, they don't always get diffusion of the media into their center, and this can cause cellular stress and hypoxia, and ultimately they can have this necrotic core. However, there is research now into tissue bioengineering to improve this, and ultimately, hopefully, this limitation will be able to be overcome. Organoids do lack vasculature, which is a problem when we think about how important the blood system is for providing nutrients to the brain. However, again, bioengineering platforms are looking at introducing vasculature to the organoid models. Some organoid models do lack specific cell types. For example, a cell type called microglia is really important for immune function, and unfortunately, a lot of the models don't express microglia. But again, there are studies that are improving this to have models that do comprehensively explore different cell types. It's important for researchers to note that there can be batch effects with organoid generation, and this is a problem. So we need this very stringent quality control to ensure that the variation in the efficiency of differentiation of organoids is low and that we have consistency in our models. This research is highly expensive to establish and maintain, and there is also a high cost of downstream experiments such as single-cell sequencing, which is really important to look at the cell type, single cell type and cell-specific pathways that contribute to mutations. Ultimately, as well, this work can be demanding and time-consuming. Stem cells require feeding every day. They are quite prone to contamination, and 3D human organoids, when generated, require daily changes in the type of growth factors, which includes work on the weekends. So how are we utilizing human brain organoids in our research? I'd like to acknowledge my colleague, Dr. Anna Cho, and PhD candidate Nicole, who've been working with me to largely transition our research from mouse studies to these human brain organoids. And we have applied all of the applications we would in our mouse models to the human brain organoids, and we've found some success with this. Ultimately, we stain the organoids to ensure that they're expressing various markers across neurodevelopment. We're able to look at their functionality by placing them onto electrodes and looking at the firing rate of neurons. And this is important to explore things like epilepsy and seizures as a phenotype of childhood dementias. So ultimately, we utilize all of these techniques to characterize and validate the models, ultimately to explore different diseases with childhood dementia as a focus. So just briefly, I want to touch on a project that was funded recently by Dementia Australia. So MAPT is a gene that is responsible for producing a protein called tau, and tau is extremely important for stabilizing our cells within our brain and ensuring that they function in a healthy way. But in dementias, the tau can become abnormally deposited within the brain and can cause the death of the neurons. Now, more recently, there have been in the literature reported cases of patients, predominantly children and adolescents, who have mutations in this MAPT gene, but more specifically, they have mutations in a chromosome region called chromosome 17 that encompasses multiple genes. And it basically deletes or duplicates a series of genes, but MAPT being one of them. These patients exhibit clinical phenotypes similar to dementia patients, and the overlap with childhood dementias as well. But most interestingly, a subset of these patients have also been found to have that same tau pathology within their brain. So this poses the question, are there links between childhood, early onset, and late stage dementias? And I'm really interested in exploring this because ultimately this rare genetic mutation overlaps quite distinctly with older dementias in that we have the same abnormal protein within the brain, the same gene is involved, and you have similar clinical symptoms. However, these are very distinct dementias. They occur at different life stages. One is quite common and one is quite rare, and the 17q21.3 one is quite a rare genetic disorder, whereas Alzheimer's disease and other dementias that exhibit tau pathology can be genetic and have non-genetic causes. So ultimately, I'm hoping this project, which will utilize human brain organoids, will be able to uncover unknown roles for really important genes and proteins within the brain across the aging process from neurodevelopment to neurodegeneration. So how we're going to do this, similar to our previous workflow, we hope to source patient lines from patients exhibiting those rare genetic mutations. But if not, in the meantime, we are genetically modifying wild-type human stem cells to exhibit these mutations. We will generate our 3D human organoids, characterize and validate the model, and ultimately perform single-cell sequencing to look at those cell type and pathway-specific contributions to these mutations. So in summary, what can we achieve by utilizing human brain organoids for childhood dementia research? Ultimately, we can explore altered mechanisms or pathways across the neurodevelopmental processes. We can also look at disease-specific features of childhood dementias, such as lysosomal dysfunction, mitochondrial dysfunction, and lipid accumulation. And because there are around 100 plus rare genetic causes of childhood dementias, we can explore each of these in detail within our models. Ultimately, 3D human organoids are greater human models of childhood dementias, and we can utilize them in various different ways and apply them to a great set of techniques to be able to look not only at features such as epilepsy and seizures, but other types of genetic and underlying pathway causes. And ultimately, the hope for these models is that we can develop drugs via our high throughput screening. So you can have a plate full of various organoids, and we can treat them with maybe even some FDA approved drugs that are already out there. And the idea would be to develop targeted and specific therapeutics that then could be safety and efficacy tested in a human context. So thank you for the opportunity to speak today. I'd like to thank the Childhood Dementia Initiative, Dementia Australia for the funding, and the Dementia Research Centre at Macquarie University.
Thank you, Fiona, for such a great overview of brain organoids and the work that you're doing. Our next speaker is Associate Professor Wendy Gold, who's head of the Molecular Neurobiology Research Group at Kids Research, the Children's Hospital at Westmead, and she's an academic at the University of Sydney. She currently leads a large team of 16 PhD students, honours students, research assistants, and postdoctoral scientists, with a research focus on developing effective treatments for rare and ultra-rare developmental epileptic encephalopathies. Thank you, Wendy.
Thank you, Chris, and thank you so much for inviting me to give this talk. It's a real privilege to present today. by way of introduction, and thanks for that introduction, I'm also a neurodevelopmental.
Sorry, Wendy, to interrupt. We can see your, it's in the presenter view.
Oh, right. I can't get it out. There we go. Let me try again. Share. Is it sharing now?
It's sharing, but it's not in present. Yeah, that's it.
There we go.
Okay.
And can you see presenter mode now?
No, that's good.
Okay, great. So what I wanted to say was, by way of introduction, I'm a neurodevelopmental biologist. And for the last 15 years, I've been spending a lot of time working on neurodevelopmental disorders, in particular Rett syndrome. And this falls under the umbrella of childhood dementias. And ultimately, what I'm interested in is how the mutations of these disorders lead to disease and how they affect the formation of the brain throughout neurodevelopment. And historically, our lab has been studying neurodevelopmental disorders in mice and in cell lines as well. However, we're now asking ourselves whether we can study these disorders and brain development outside the embryo, and in fact, whether we can use these 3D organoids. And if this is possible, then we can start to understand more about the developing brain and some aspects of the developing brain using cells from patients that have childhood dementias. And these emerging models are very important because this is the first time that we can begin to understand the development of the brain and how disease actually affects the developing brain. Now, the part of the brain that we're most interested in is the cerebral cortex. And this part of the brain contains many, many different cell types that interact together to ensure normal brain function. And why we're so interested in this region of the brain is that the cortex develops during embryonic development, and it continues to develop over a long period of postnatal development, where experience-dependent maturation of neurons and circuits occurs. And the reason why the cerebral cortex is so important is it coordinates and it controls really complex functions of the human brain, such as thought, movement, language, sensory perception, memory, and cognition as well. But it's not that easy to understand how the brain works, and especially in the context of disease, as access to brain tissue is very limited. And in addition, as good as mouse models are, which Fiona alluded to, and as good as they are to helping us to understand brain development and brain disease, their brains are just very different to humans. And so there's been this enormous drive in the field to develop new models, which are human-like, such as cortical brain organoids, that allows us to model human brain disorders, such as childhood dementias. And using brain models, such as cortical brain organoids, we can start to ask these very important questions about the developing brain, and specifically whether the cerebral cortex and other brain regions, for that matter, can be used to model the human brain and model development of the brain, such as those found in childhood dementias. Brain organoids, as Fiona was saying, were just essentially just a cluster of neuronal cells that have many characteristics of the developing brain. So how can we create these human cerebral cortex organoids? Well, first, we can take blood or we can take skin fibroblasts from patients, and we can reprogram them into what we call induced pluripotent stem cells, or iPS cells. From these cells, we can then grow them into any cells that we want. And in our case, it's neurons, because we're interested in neurons. And this is a very well established pipeline. And through this well-established pipeline, we can then start using different reagents and cues to form neurons and organoids. We can then start to model the brain disorders. Now, with these models, we can then ask questions about how the human brain develops and how the mutations that cause childhood dementia can affect the function of the brain. And then we can use these, as Fiona was saying, to test for novel drugs. It could be FDA or they could be novel, and also screen novel therapeutics as well, and also look for biomarkers. And the exciting aspect of this is that we can test these novel therapies and drugs and look for biomarkers on these organoids that come from the very patients that have childhood dementias, thus allowing us for more precision medicine approach. Childhood dementias are a devastating group of disorders, which are neurodegenerative, progressive, and severe in nature, and many do not live into adulthood, with some children dying in their infant years. Childhood dementias are an umbrella term, which consists of more than 70 different genetic conditions, where only 5% have effective treatment options. Rett syndrome is one of the most prevalent childhood dementias, and this is one of the large focus of my research areas. And Rett syndrome is largely caused by pathogenic variants in the MECP2 gene. And like most childhood dementias, there's currently no cure. Diagnosis is clinical, and treatment does remain symptomatic. Now, so in collaboration with the Stem Cell and Organoid Facility at the Children's Medical Research Institute, which is led by Dr. Nay Gonzalez-Cadero, we've been growing cortical brain organoids derived from induced pluripotent stem cells from Rett syndrome patients. And by now we have about 10 different patient lines and their controls. And using specific growth factors and conditions, we can grow these 2D induced pluripotent stem cells, which you can see here, and after some time, they then reorganize themselves into these 3D ball-like structures, which we call brain organoids. And if we use specific antibodies against cell-specific proteins, we can start to understand the cellular composition of these models. And here you can see the induced pluripotent stem cells growing in a dish, and here are their markers of pluripotency, which include OCT4, NanoG, and SOX2. And then if we look at the cellular composition of the brain organoids really, really early on in culture, so day 25, we can start to see cell types emerging, such as neuroprogenitor cells and neuronal neuronal stem cells, which are marked by PAX6 and SOX2, and DAPI stains the nucleus. And you can see this beautiful patterning early on in development at day 25 as the organoids develop. And then as the organoids get older, these organoids have been in culture for 60 days, they start to, you can start to see cells such as neuronal cells, which is marked by MAP2 and TUJ1. And you can see the different layering in these organoids. And if we overlay them together, you can see these nice cellular processes and the interplay between these neurons. And then we can start to see the introduction of astrocytes using a marker against astrocytes, antibody against astrocytes called GFAP. And as you can see here, at 2 months old, when the organoids are 60 days in culture, we can't see the astrocytes, although we can see the neuronal cells and then DAPI stained in blue. And then at 4 months of age, where the organoids have been in culture for 120 days, we can now see the astrocytes and marked increase in expression of MAP2, and this is the overlay as well. Then we can start to see the interactions of these cells. If we take a deeper look into these organoids, when we zoom into these regions here, we can see that the astrocytes are marked in green and the neurons are red. And if you look closely here, more zoomed in, you can actually see the astrocytes and the neurons together in the organoid. Also, what we were interested in, we were very curious to know what type of neurons were being expressed in these organoids. So we used antibodies against excitatory and inhibitory neurons, VGAT and VGLUT. You can see here that the cells express VGAT. These are this is a neuronal marker, VGLUT as well in the overlay. So now we know that these organoids, they're expressed neurons, and we can actually understand more about what type of neurons are being expressed. We know that they're excitatory and that they're inhibitory as well. And interestingly, we can also see some neurons that express both excitatory and inhibitory markers. And so this demonstrates this really complex mix of cell type in these organoids. So just to orientate you, MAP2, which is a marker of neurons, is in purple, V-GLUT is in yellow, V-GAT in a cyan, and DAPISAN's a nucleus. And this is the organoid, and this is another deeper dive in, and then an even deeper dive in, so we can see exactly what type of cell types are being produced by these organoids. And finally, we can demonstrate these neurons actually have activity and they can communicate with each other. So using a fluorescent calcium ion sensor, we can actually demonstrate neuronal activity and the dynamics of neuronal networks in the organoids. On the left-hand side, you can see the activity of a control organoid mimicking the activity and network of these cells in the brain. And then on the right-hand side, you can see how neurons in a Rett syndrome organoid, so this will be from a patient that does not express MECP2 or functional MECP2, and you can see that they're not very active. And so this demonstrates the functional effects of MECP2 deficiency in these organoids. So finally, just in conclusion, hopefully I've convinced you that patient cells can be reprogrammed into stem cells, which can be differentiated into cortical brain organoids. And cortical brain organoids have many characteristics of the developing brain. I've showed you here that these serve as a reductionist model of human brain development because they're not the whole brain, they're just part of the human brain. And cortical brain organoids can help us answer some really important questions about the developing brain and how childhood dementia affects the development of the brain. And so lastly, I'd just like to acknowledge everyone that's helped with this work, especially Jasmine and Kelly and Eleni from my team who have produced all these wonderful images and done all this amazing work. And then of course, Dr. Nay Gonzalez Cadero from the Children's Medical Research Institute, and my funding sources as well, and also my partners as well. Thank you very much.
Thanks so much, Wendy, and thanks for those beautiful images and videos. That was really, really impressive. Thank you. So our next speaker is Dr. Brett Kagan, who is the Chief Scientific Officer at Cortical Labs. Cortical Labs is a multidisciplinary deep tech startup looking at integrating hardware, software, and synthetic biology approaches to explore how to harness intelligence from neurons on a chip. Dr. Kagan completed a PhD in neuroscience, focusing on stem cell therapy, and completed postdoctoral work in bioinformatics and regenerative medicine. Thank you, Brett.
Thank you so much for that introduction. You can all hear me okay, I hope. Someone shout out if not. Today, I'm going to be presenting some stuff that in many ways is supplementary to the really awesome work that we've seen so far, but might help in terms of the actual modeling and how we can proceed with that. So I've titled this talk to model the thing itself. In other words, how can we assess in vitro neural function? And when you get down to it and you consider something as complicated as a brain, we heard brilliantly before from Fiona about the comparisons with the mouse. You know, the nice thing with a mouse, even though it doesn't genetically match us and the brain is different, is we can always see how does its brain work, because we can give it little cognitive tasks and see how that happens. But that's very, very hard in vitro. Even though we can look at proteins and molecules and activity, there's still a lot going on there. And so we became really interested in this idea of, can we try and combine all of these different areas of research that's been done to try and interrogate deeper how processing happens within the brain? And so we brought together different advancements in hardware, software, neurocomputational understandings, and synthetic biology. And we're interested to see, you can see here in the figure on the right, this idea of this little simplified game of Pong. Could we actually train neurons in a dish to play the game Pong? And so there were three key parts that we sort of can think to break this down into. One is to improve some hardware and software to allow for us to create this closed loop implementation to provide the information to the cells and read information from the cells. The other one was, is there actually a theory we can use to actually motivate cells to behave in any particular manner? If we want to look for learning, we need to motivate them to see why learning or a subset of it might occur. And then finally, how do we actually produce the cells? And we've heard some really brilliant stuff already today about some really great synthetic biology approaches to creating these models through organoids. I'm going to go really high level and fairly quick on this so we can get to some of the other stuff. The first step we had to do was create what we called an embodied neural network. Now, if that sounds fancy or whatever, it's not as complicated as it sounds. Essentially, you can think of there being two broad feedback systems. One is an open feedback system, where you put information into, say, the neurons, they have a process and you read that process out. That's an open feedback system. And that is almost all, there are a few exceptions of other people like ourselves doing this work, but nearly all the work is done in this manner, where people do this open loop feedback. A closed loop feedback system isn't that much more complicated. The only difference is you close the loop. You take whatever that output is and you use that output to change future incoming information as a form of feedback. And you and I and everyone watching this all live inside closed loop feedback systems. As we move, we feel the movement. As we pick up a pencil, we can see it be picked up or dropped or whatever the case is. So we live in a closed loop feedback system. So if it's complicated, just think you need to be able to create a system that you can respond with. So all it means is you have to create a sensation, measure the response, and then provide a sensory feedback on that response. And ideally, it should be, if you're trying to communicate with neurons, high resolution, real-time, physiologically relevant. But even if you're able to achieve this, you're still left with the question, why would a group of neurons actually organize their behavior in a changing environment in any particular way? You and I have really distinct reward pathways. We have biological incentives. We need to eat, do several other actions to survive. But why would a group of neurons do that? And so we had to find a really high-level theory. And I don't have the time to explain that in detail now, but we're adopting something called the free energy principle, which basically proposes that all neural systems try to minimize the difference between an internal world, which is a model of the world, and the external world to minimize the gap between these two models. So again, if we use the example of myself, I have a glass of water, but what I operate is not truly on the physical world. I operate on the model of the world my brain has developed. The implication from this is that if there's something that cannot be predicted at all, something that is truly random, that might be aversive under this model, while a predictable re-stimulation, something that's very consistent each time, might be reinforcing, because it's a way that the system would minimize this free energy, and that the neural system would modify their activity. So what we did was we said, all righty, we'll do a simple game of Pong, we'll put some information in, and then if they get it wrong, we're going to give them random noise as feedback. And if they get it right, we'll give them predictable feedback. And so then the final step we had to do was culture cells. Now, in this particular work, we did not culture anything as sophisticated as the organoids that we were showing today. Instead, we started out with much, much more basic systems, either from primary mice, embryonic mice as a very standard, or using an iPSC-based approach, very similar to how the organoids were generated, but simpler, in these cases, just monolayers. And we were able to compare these cell types and plate them on these multi-electrode arrays, which is basically a special type of chip that can record the electrical activity from cells and provide stimulation to them. And you can see here some of those cells growing on a monolayer on a multi-electrode array, and they're able to survive for upwards of 6 months, at least in the human cells, fairly regularly. And this is a little map of the electrical activity as different cell types matured at a different stage. But the important thing is that they all matured and showed this robust electrical activity. So finally, what we did was we brought all of these different things together. We provided the structured information that's actually most similar in quality to what a rodent barrel whisker cell is, if anyone works with animal behavior, well, not animal behavior, but animal research in that area. But also has a link to sort of how you would topographically map information in your own brain as you receive it visually. And then we made sure that this loop was very, very tight with about a 5 millisecond latency. And that allowed the neurons to alter the position of the paddle and then receive updated information about where their paddle was in relation to the ball. And you can see a little quick video here that describes what that looks like. So you had either sensory or motor regions being counterbalanced as per the previous image. each of these bars is electrical activity from neurons. This is 2 times the speed. And then as they change their activity in those 2 counterbalanced motor regions at the bottom, so it's sort of left, right along the bottom, they were able to get better at playing the game of Pong. And that was quite exciting for us to see. And we quantified it here with a number of different control groups. And what we found essentially was if you compared, say, a media-only control to, say, a mouse cortical cell or a human cortical cell, what you could see is very distinctly that by the latter part of the gameplay, they were getting better at hitting the ball. And then we also compared other things like in silico computational models as a control, and REST, where there were cells receiving information, that's RST, where the cells were receiving information but not actually playing the game. Sorry, they weren't receiving information, but their activity was moving the paddle. Sorry, it's kind of like blindfolded as a bad analogy. Sorry, I've mixed that one up. And none of them showed performance. And you can see that very clearly here in these box plots. And while there is significant variation, nonetheless, the control, the in silico control, the REST controls did not show any learning, while the mouse cortical cells and the human cortical cells did show learning. But this work's all kind of old work now, and we published it, and I presented to you just to sort of showcase the model. And if you're interested, there was a paper published in Neuron last year that discusses this, and another few follow-ups that have come out this year that you're very welcome to go and explore. But our conclusion from this essentially was that we're able to use this dish brain system to get this very basic intelligence from these neurons and test them. Now, the question might be, why is this useful in the context of what we're talking about today? How can this actually help us understand what might be going on in disease or in testing drugs? And quite simply, although we've seen, again, I won't go into it in detail because it's been presented so well by Wendy and Fiona, that there's advances in iPSC-based cell cultures, and that can give you access to patient-specific biological material, there's still really high failure rates when you take that to the clinic. And at the end of the day, that's because what people are looking at is these proxy markers. And whether that's protein binding or firing rates or some other marker, the true function of a neuron is to actually integrate and do something with information. And that can be incredibly complicated information that, say, for a human cerebral cortex is able to integrate, or it can be more basic, like something occurring in the brainstem, but it's still a way of transmitting information around. And so our hope is that if we use this synthetic biological intelligence to provide an interactive environment, we might actually be able to better understand what's going on. And so we asked the question, could we model a disease? For example, here we're interested, could we model epilepsy? Epilepsy is quite well understood in a dish. There's a number of fairly robust epilepsy models for drug screening in a dish. And so we grew this excitatory type of neuron, and we tested it for activity and gameplay over time. And then we also applied anti-seizure, also previously called anti-epileptic medication, at different doses, and investigated what was going on. And what we found was that not only were these cultures showing evidence that they had this hyperactivity often seen in epilepsy, some epilepsy phenotypes at least I should say, they actually showed evidence of being a drug-resistant epilepsy model. And so you can see here the cultures showing their dense interconnectivity and their very, very dense activity in the vehicle control. So these are the epileptic cells. So it's just constant activity all the time. And then when we applied an anti-epileptic or anti-seizure medication to it, we're actually able to reduce that activity and achieve more discrete rhythmic behavior. And as mentioned, we tried a number of different anti-epileptics, including phenytonin, perampanel, carbamazepine, and we found that only carbamazepine was able to reliably reduce the activity of these cultures. Some of them, like perampanel, for some reason, mechanism of action we're yet to identify, actually increased the activity. And so we decided to focus on carbamazepine and found that actually what we got was, compared to controls, a very consistent and reproducible improvement in performance in this still using Pong game, but in a dose-respondent use of this with this particular anti-seizure medication. And so that's really exciting for us because it shows not only do a disease model fail to show learning, as one might reasonably expect, but you can, to a small extent, not as much as what we saw with our fully healthy cultures, but a small and statistically significant extent, rescue that learning with a drug designed to treat that particular disease phenotype. And so that was an incredibly exciting thing for us. Now we're a bit short on time, so I'm just going to flick over and flag very briefly that we've done this looking at aging cell lines as well, and found a number of different nuances, looking at basic activity, some gameplay, again, more moderate results than what we see with healthy lines, but more importantly, looking at things like neural avalanches or neural criticality. This can be pulled out and represented in a disease model. And so we can go very, very deep into understanding exactly how these systems are working. And for us, we have a lot of broad expertise, but we don't have a deep expertise. And so our core focus has not been, can we use this tool ourselves to answer all of the problems that might be out of there, because just so many of them, but can we improve the accessibility of it? And we do see this area very similar to the nascent computer industry. You know, previously it took a team of PhD qualified people, you know, in a room to run it. You know, if anyone was around for the S360s back in the 60s and 70s. And that's kind of where we're at right now with the laboratory setups to culture cells. I mean, you've seen the amount of work that people have done to culture these stem cells, grow them into organoids or monolayers and use them. But the big breakthrough with traditional computing wasn't people continuing in the lab, you know, where it takes a whole team to run it, but it was making it more accessible so more people could do more stuff with it. And that was really what sort of stuff like the Apple II and the personalized computing revolution achieved. And that's where we want to be. We want to try and introduce a way to make this more accessible for people, so you don't need an entire team to use it. And so we've been building these perfusion circuits that actually maintain the cells over a long period of time. And I think you heard, I think it was Fiona was talking about, it requires a lot of weekend work normally to keep organoids alive. Hopefully this should reduce that work a little bit. And this is actually the new version that we now have that we're piloting and we'll be going to production with to make accessible. So it will integrate everything, including a multi-electrode array reader inside it. And the goal is to be able to expand up and be able to do this in a high throughput manner. And along with that, we're going to be introducing a cloud-native web-first development tool. And this means that you can actually be able to build up environments. So for us, that initial Pong environment I showed you took about 18 months of incredibly talented programmer development. And in truth, it was still very janky. It's still very much, and we've been really upfront about that, like a very much a proof of concept that you can, in fact, convince neurons to do something to some degree, but there's so much more work that could be done. Here, you can code a similar environment in a matter of days, a couple of days, even if you aren't a highly skilled programmer. And so really, again, it's about just making this entire process accessible, even over the cloud or in the lab. The idea is just to make it easier to do these sorts of investigations than what we've currently been done. And I see I'm out of time, so last slide. Really asking the question, where does that leave us? When we're looking at stuff that's really trying to progress into the future, whether it's developing these really complex organoid models to capture these nuanced disease states, or whether it's trying to interrogate them, I think one thing all the researchers can agree on is that there's a lot of uncertainty in how to move forward, but also a lot of promises. And while there are many, many challenges to really harness the full potential of these technologies, and there is significant potential, I think, as you've seen from all the talks today, many people sort of look at it and say this idea still sounds like science fiction. But I always like to end the talk and saying that science fiction ends and science begins once we start working on the problem. And the people here today are all working on the problem. And our hope, what we hope our small contribution can be, is that if we can enable a community-wide effort to going down deeper into how we investigate this problem, we might have a greater chance for success than any of us just working alone. And so with that, I'll just end up and thank my amazing collaborators and team that we have working with us. And again, if anyone has any collaborations, would like to work out, would like to explore these ideas with us, please do get in contact. My email's there by the bottom. Otherwise, feel free to follow us on the social medias. Thank you very much. Back to you, Chris.
Thanks, Brett. That was great. Really interesting. Thank you so much. If we can get all of our speakers to turn their cameras back on. And if anyone has any questions, please feel free to write them in the chat. I can't see any questions there yet. Does anyone in the panel have a question for each other?
Happy to throw a question over to the other researchers and to Stefan if he's seen anything in this area. I'd just love to know if people think that there are any particularly interesting phenotypes that have been expressed so far that merit unique investigation into the pediatric dementia pathology that you've seen in vitro compared to, say, like other forms of dementia, anything that really stands out regarding the pediatric aspect?
I think one thing I could throw into the ring is I think in some of the childhood dementias you have over-enthusiastic synaptic pruning that can I think could be involved in some of the phenotype.
I think I mean, we're just slowly transitioning the patient lines and that into the organoids now, but I think what's quite evident is that they're very sick, the organoids, from a very early on stage, which obviously just demonstrates the severity of these conditions, and how that maybe working at certain time points are going to be very critical, because maybe really early on is not going to be feasible for a lot of these models, because they're just not going to survive. So yeah, I think what I've been seeing is some very unhealthy looking cells, which obviously shows that the disease is there and something is happening. But getting them to survive through the models might also be a bit of a challenge for the more severe diseases as well.
I've got a question pop up from Lisa Melton. So she says that Fiona briefly touched on this, but could Fiona and or Wendy discuss the potential of seeding organoids with microglia for a more full picture of cell-cell and organoid drug interactions?
Maybe Wendy, if you
We don't do that, but it is a great idea, and definitely something that that is very important for the research as well. Specifically for childhood dementias or any brain disorders where the microglia are so important and they're involved in the pathology. So this is definitely something that the field is working on in order to include microglia. As Fiona touched on as well, it's also important that we look at new ways and try new ways in which to introduce vasculature, or at least endothelial cells that can support these neurons as well, because the inside of the organoids have this necrotic core. So all the cells that are in the brain are all very important. And I think as the field progresses and grows, I do believe that the field will advance and more of these cell types will be able to be included so that we can have a more representative model.
I've got a question here from Natalie Anderson. She says, excellent presentations. Thank you all. How easy do you find getting funding and access to seeding cells for organoids in Australia? And what do you see as enablers for the acceleration of this amazing technology in Australia? We always need more funding for this research, don't we? That's a given.
We all know the situation of funding in Australia, whether it's for using mouse models or organoids. But you know what I wanted to say is that one of the most powerful enablers is the families and the communities. And without them, we wouldn't be able to have these samples. So having the families and communities invested in this research and then getting their consent is really, really important. We do, I think Fiona touched on this, develop some lines where we use CRISPR gene editing to introduce a mutation or to correct it as well. But this is not as powerful as actually having a cell line coming from a patient.
Absolutely.
I would also, if I can extend on that, because we got our funding from a very, very different source than the academic route. I would say for innovative therapies, the funding model in Australia does not work very well. There are a few notable exceptions, and I think the people who have managed to leverage the existing systems to achieve stuff are amazing, because grants and the current way of applying for funding is very, very tough. And so I think it's just like amazing for those people who have achieved stuff and managed to do a bit of work through it. We never would have got, I don't believe we ever would have got funded to do the work we did from the pure granting system. And so I think it would be brilliant to establish better pipelines for researchers to access other forms of funding, whether that's, philanthropy from wealthy investors. There's a lot of wealthy investors, and I know that like people like the work that Stephen's setting up is so important to try and bridge the gap for researchers to those other investors, but also like consider where work can be taken commercial and strike out from the university where possible, because there's a lot of money out there and a lot of people who want to see good done, but it can be tough to do it through a pure university setup sometimes. So, yeah.
Yep. So we've got time just for one more question. So this one's for Brett from Carissa, and she says, congratulations, some of the coolest neuroscience I've ever seen. Sorry if I missed this, but are your cultures pure? Are there any glia popping up in there? Could the variability be attributed to this? And also, do you have any comments on how far you can take your work from an ethical standpoint?
Yeah, absolutely. Great question. We normally have a slide. I just cut it out today because I had a feeling you were going to see a lot better immunos from the other researchers than myself. But we did characterize them. We do have glia cells, no microglia yet, as far as I'm aware, but we did have other supporting cell types. We culture both pure and not pure cultures, and I think this touches on a bit what Wendy was saying as well, is that brains are incredibly complicated. And when we try to build up cultures using pure populations, so let's say like just pure glutamatergic, pure GABAergic, some astrocytes, combine them in different ratios, we get to see compelling results to capture things. There's a lot that goes on in the brain, and it's an ongoing work for us to try and identify what we often call like a minimal viable brain is, an MVB, to actually get a learning effect out. So that work's ongoing, and definitely you need a lot of different things in there to get it to work. Regarding the ethics, we're really committed to engaging with the ethics. I'll put a link in the chat here. We recently just published a work with a number of different independent philosophers and bioethicists actually tackling the challenges and opportunities that this field raises, and making sure that we can maximize the ethical benefits, such as maybe where possible reducing animal testing, getting access to therapies quicker, while actually reducing the concerns that are around it. So we're very well aware of that, and the paper's in the chat if you would like to. Oh, that chat might be, sorry, that was just to one person. Yeah, to the group chat. So that paper's there if you'd like to have a look.
Great, thanks. We can circulate that as well. So we're out of time. We've actually gone 3 minutes over. And I'm sorry for those people whose questions we didn't get to, but we will circulate those questions and get an answer to you. Thank you so much for joining us today. As always, feel free to email me with any questions. My email is on the screen there now. If you haven't already, join the Research Alliance and have a look on our website using that QR code for all the stuff that we can provide to you. And look out for information about our next webinar, and we'll let you know about that as soon as we can. Thanks all. Enjoy the rest of your day.